My name is Mårten Rooth. I am the CTO of Nanexa and one of the co-founders of the company. My background is in chemistry, and I have a PhD from Uppsala University in materials chemistry. In my role at Nanexa, I've been part of developing the PharmaShell process and technology, and I've been doing so from the very beginning. And I've also been part of building our GMP and production capability. I mean, there is always a balance between complexity, added cost for a technology compared to the opportunity it might give or the improvement of that technology. With the PharmaShell technology, I believe that the improvement far outweighs the added complexity and the costs. And when it comes to scale up, in fact, our technology could potentially avoid one of the major scale up challenges, which is the aseptic fill finish. And we can do this because the PharmaShell technology enables terminal sterilization. That is, that we can sterilize the product in its final package. And we can do so over even for sensitive drugs like peptides. And that is something that not many other technologies can do. I would say that it fits perfectly into how the market for type 2 diabetes and obesity is evolving, with less frequent dosing being a very hot topic. Our ability to work with virtually any GLP-1 molecule, old ones as well as new ones, makes us competitive. And our technology is actually not limited to just once monthly depots. We can do much longer depots than that. And I think that could be very attractive also when it comes to maintenance treatment and similar. With our technology, we can make long depots. We can make really long depots. And it's regardless of the half-life of the API that we work with. So even for the new APIs coming that have longer depots, and I mean, Pfizer bought Metsera basically for their technology platform when it comes to this GLP-1s. And they have a really, really long half-life. It's, I think, 16 days, and it's the longest one I know. But even for that product, we could actually extend the duration of that product. And I don't know how far they can take it by working with the molecule, but we leave the molecule unchanged, which is also an advantage. And we can make really, really long depots. And we also protect the API with our technology, which is important if you're going to leave, let's say, if you do a more quarterly dosing, then you really need to protect the API when it's in the body as well. Otherwise, it will deteriorate there and you don't get any effect, or you need to administer quite a lot. This protective property of the PharmaShell also helps when it comes to the production, because with our technology, we can use terminal sterilization. And that is because the PharmaShell technology also protects the API during sterilization. So we can work with terminally sterilized product. We don't need this very this bottleneck process of aseptic fill finish, which is one of the major contributing factors to the supply chain problems that both Novo Nordisk and Eli Lilly experienced this and last year. And also coming in from the protective properties, we have seen that we can store these peptides at room temperatures after sterilization. And instead, so we don't need the cold chain logistics, which is a significant cost actually for biologics. So that is a couple of advantages. That and also that with our technology, we can make really highly concentrated suspensions, which means that the injection volume is low, which means that we can also use very fine needles. So it's very convenient for the patient, which is very important in this market because it is so much centered. It's a consumer-driven market, which is maybe not the most usual when it comes to pharmaceuticals. We like to be one step ahead when it comes to scale up and production capability, so that we can match the results that we have achieved for our long-acting injectable GLP-1 project, NEXT22. And CPHI meeting is one of the better places to meet and keep up to date with existing and new suppliers and partners for production, who are very important for our readiness to be able to produce larger quantities so that we can supply both our own future studies and also our partners.